नाज़रीन, कश्मीर लाइफ के साइंस टॉक्स में आपका खुशामदीद। जैसा कि आप जानते हैं, हम इस टॉक शो में काफ़ी टाइम से कश्मीर के साइंटिस्ट से बात करते हैं, उनकी लर्निंग कर्व जानते हैं। तो आज की इस कड़ी में हमने दावत दी है जनाब डॉक्टर एजाज़ हमीद वानी साहब को, जो बायोटेक्नोलॉजी से हैं। इनका जो पूरा काम अभी तक रहा है, रिसर्च कर रहा है, वो ये जो जीनोम है, ज़िंदगी की जो बुनियाद है, उसको स्टडी कर रहे हैं काफ़ी टाइम से। तो आज हम इनके जीनोम के बारे में, जीनोम के चैलेंजेस के हवाले से डॉक्टर साहब से बात करेंगे। हम शुरुआत इस बात से करेंगे डॉक्टर साहब, एक ज़माना वो भी था कि जब हम इस तलाश में थे, कायनात, ये जो दुनिया इस तलाश में थी कि ये डीएनए जो है तो है क्या। अब तो डिकन्स्ट्रक्ट हो गया है, अब तो पढ़ा भी जा सकता है, डिकोड भी किया जा सकता है, इसकी लंबाई भी जानते हैं, सब कुछ जानते हैं। लेकिन फिर चैलेंजेस क्या हैं हम?
सो स्टिल एज यू मेंशन, जैसे आपने कहा कि हमने बहुत कुछ जाना है इसके बारे में, बट आई वुड से दैट अभी व्हाट वी नो इज़ जस्ट स्टिल अ टिप ऑफ द आइसबर्ग अबाउट द जीनोम। जो जीनोम डीएनए के बारे में हम अभी भी जानते हैं। सो जितना जीनोम के बारे में हम जानते हैं इज़ लाइक 2 to 3% अभी हमको सिर्फ पता है कि क्या उसकी क्या फंक्शन है, वो क्या करता है। रेस्ट अबाउट 95%, 98% वी स्टिल डोंट नो व्हाट इट डज, व्हाई इज़ इट रिक्वायर्ड, इसका क्या फंक्शन है। ठीक है, सो इट इज़ स्टिल लाइक अ लॉन्ग वे टू गो फॉर अस के जैसे हम इसको फुल्ली अंडरस्टैंड करें, बिकॉज अभी हमको बस यही 2-3% का जीनोम जो कोड करता है, जो प्रोटीन्स बनाता है, ठीक है, जो हमारे बहुत सारे फंक्शंस फिजियोलॉजी ड्रिवन करता है, इट इज़ जस्ट 2 to 3% जो हमको पता है। आल्दो हमको पूरा जीनोम हैज़ बीन सीक्वेंस्ड, लाइक हमको पता है कहाँ कहाँ पे क्या क्या ये है, इसकी बेसिस है, सीक्वेंस हमको पूरी पता है, लेकिन उसकी फंक्शन क्या है इन द ह्यूमन फिजियोलॉजी, इन द ह्यूमन डेवलपमेंट, वो हमको अभी भी पूरा इल्म नहीं है। एंड इट विल टेक अ लॉन्ग, वेरी लॉन्ग वे टू अंडरस्टैंड दैट।
तो ये पढ़ने में जो आया है वो कि ये जो डीएनए का स्ट्रैंड होता है, वो चाहे प्लांट लाइफ ले लीजिए या आप एनिमल लाइफ ले लीजिए, एक जैसा है।
हाँ,
तो फ़र्क क्या है?
सो फ़र्क ये है कि डीएनए आप समझो 4 मॉलिक्यूल्स का बना है, ठीक है, और हम उनको बोलते हैं ATGC बेसिस। सो द वे दे आर ऑर्गेनाइज, जो इनकी सीक्वेंस है, जो इनकी तरतीब है एक के बाद एक के बाद, वो वेरी होती है। देयर आर इनफाइनाइट नंबर ऑफ पॉसिबिलिटीज बाय विच वी कैन अरेंज दीज 4 बेसिस, which makes plants a plant and which makes a human being and a virus a virus. It is only, I can say, the sequence जो इनकी तरतीब है इन 4 मॉलिक्यूल्स की, वही सब डिटरमाइन करता है कि what will be the organism, what will be the fate of the organism.
दुनिया बहुत बदल गई है। एक ज़माना था जब कंप्यूटर नई चीज़ थी। आज तो ये जो हमारा मोबाइल हमारे हाथ में होता है, वो तो क्लासिक कंप्यूटर है। एवरीथिंग इज इन दैट। अभी आप 1000 कंप्यूटर इकट्ठे जमा कर लो तो सुपर कंप्यूटर हो गया। अब तो ज़माना क्वांटम कंप्यूटिंग का है। तो इतनी सारी फैसिलिटीज़ होने के बावजूद अभी भी चैलेंज है हमारे यहाँ।
हाँ, बहुत सारे जो हमको पता है, जैसे मैंने आपको बोला कि ह्यूमन जीनोम हमको पूरा पता है। No, it's not only human genome, but the genome of the hundreds of organisms, plant हो, microbes हो, viruses हो, human beings हो, cattle हो, हर एक की application है। When I say genome, I mean the sequence of these 4 molecules. Okay, so हर एक की पता है। So it only happened because of a technical revolution. Okay, जब पहला human genome sequence किया गया, so it happened at Broad Institute in Boston near and MIT. So and people spent billions of dollars, was funded, NIH spent, जो US की funding agency है, उन्होंने billions of dollars इस पे खर्च किए to sequence that. But आज मैं अभी बता रहा हूँ, इस लैब में हम बैठे हुए हैं, I can take my genome, I can take your genome and get it in sequenced within a week and give it, this is your sequence. This is the sequence of your genome within a week and at the cost of about $1.5 lakh. So this is a huge technological revolution. about knowing the genome of an organism. So कहाँ billions of dollars just few decades back and कहाँ आज 1.5 lakh यहाँ हम बैठे हुए हैं यूनिवर्सिटी ऑफ कश्मीर में बात कर रहे हैं जीनोम के बारे में, we can get it done. So this is a huge technological revolution which happened and which enabled us to gain the knowledge that we have right now. So this is in terms of knowing the arrangement. We have 4 bases का sequences in the genome. But now, as I said, the function of it. ये क्या करता है? अगर ये स्ट्रेच है डीएनए का आपकी सेल में, हमारी सेल में, या प्लांट की सेल में, या वायरस की सेल, इसका क्या रोल है इसकी फिजियोलॉजी में? So that is what we still don't know exactly. Okay, so how it happens? Okay, what is its function? So that is going to take some time more. Okay, but as I said, there was a huge technological revolution which helped us to sequence, know the arrangement of these bases in hundreds of organisms.
ये तो सुपर सब माइक्रोस्कोपिक दुनिया है.
Definitely.
इस पे तो बात कर लेंगे, लेकिन जो दुनिया विजिबल है जिसमें आपकी लर्निंग, आपकी एक्सपीरियंसेस, आपके तजुर्बात स्कूल से लेकर जहाँ इस लैब में हम बैठे हैं, वो एक लंबा सफ़र है, बहुत विजिबल है. थोड़ा हमें भी बता दिए, हमारे नाजरीन भी डॉक्टर एजाज साहब को समझ लेंगे.
So yes, it has been quite a bit of journey. So I am from Daksum, Anantnag, which is I think that probably the last village
on way to
Kishtwar. Okay, so my initial schooling happened in that local village, in a school there, and then I did my BSc in the government college, Anantnag, and I did masters in biochemistry from this university in the department of biochemistry. And there I moved for PhD to National Center for Biological Sciences at Tata Institute of Fundamental Research in Bangalore. Okay, and where I worked on protein biophysics. That was my PhD. And then I moved for my postdoc to Harvard University in 2009 and was there for about 5.5 years. Okay, that's where I switched my field and tried to understand the genomics and the packaging of the genome within the cell. Okay. And then we got these transitional fellowships, which is Ramanujan Fellowship, and I came back and joined the Department of Biotechnology at the University of Kashmir. So, and then obviously, as we are talking right now about what we are doing in the labs, and we set up an individual lab at the Department of Biotechnology, okay, and CIRI, as you can see here. And for that you need to have a lot of funding, okay, because as you know, the equipments which we have in the lab and which we have the reagents, the other things, the chemicals which we need, they cost a lot of money, which means we need to have a funding. So what I was lucky to get is one of the highly prestigious grants from the Wellcome Trust DBT India Alliance, which is the premier funding agency in the India and between the UK and the India. And they fund very few people every year, and it's a tough competition. I was lucky to get 3.5 crores, and I was the first and probably the only person from J&K to have.
In which year?
It was in 2017, which I got. So that was quite a quantum jump in my funding and my research funding to my lab, which enabled me to set up this world-class research program on genomics.
So उस पर बात करने से पहले एजाज साहब, मैं ये जानना चाहूँगा कि आपने जब PhD किया तो PhD में आपने किस बात पर काम किया और what were the takeaways from that research in जो आपने टाटा इंस्टीट्यूट में किया?
So PhD, I was in, as I said, I did my PhD in protein biophysics. So we were trying to look at the 3D structures of the proteins and how they change, okay, and how proteins sort of basically a protein can function only when it is in a 3D shape. If it is linear, it will not function. So my topic was to understand what are the principles which determine the 3D shape of an protein molecule, okay, and what's its relation to the function. So, and if we say my takeaways from that, I found that the proteins are highly dynamic structures. They fluctuate all the time, okay, like we breathe, we move a little bit, we jiggle here and there, okay, so we are resting, but we are a little bit jiggling here and there. So proteins do the same thing. They jiggle and wiggle around. I used a very high sensitive technique, which is mass spectrometry, to look into the dynamics of these proteins, and I found that the proteins are highly dynamic, and this dynamics is related to their 3D जो उनकी shape है उसमें उसका role रहता है. That was one of the major takeaways from my research. And in addition to that, what we found that the proteins, they can sort of come together, molecules एक दूसरे के पास आते हैं, they make a big blob in sometimes, which can lead to the neurodegenerative diseases like Parkinson's है, Alzheimer's है. So वो एक part रहा था उसमें, to understand the molecular basis, the dynamics, जो jiggling और wingling up proteins और molecules है उनका फिर aggregate हो जाना, एक जगह आ जाना, and then leading to a disease like Parkinson's or Alzheimer's.
तो is there some kind of मतलब कुछ कोई knowledge है हमें इस बारे में, शायद epigenetics इसको कहते हैं?
नहीं, it is something little bit slightly different. बेसिकली प्रोटीन्स की बहुत सारी चेंजेस हो सकती है जिसकी वजह से वो कभी एक साथ आ जा सकते हैं, एंड कभी समटाइम्स दे कैन गो अपार्ट फ्रॉम ईच अदर, ओके. सो एंड if they come close to each other, sometimes it is beneficial locally if they come close to each. That's what is the new concept now building up, which is called which I will put in a term, highly technical term, which is called the phase separation term. Usme proteins, what we right now say is that it is beneficial that proteins come together and they form a very specialized compartment which helps in driving the physiology of the cell. But in case of a disease or something, they become together, but their structure changes, and that leads to the pathological condition that leads to the altered physiology of the cell, which is obviously not good for us.
You continued with the same kind of research once you started your postdoc at Harvard?
No, I switched 100% to a field of which I had no technical expertise before. So that's one of the important things that I think we should look forward, or we should not be scared of not doing something which is new. So I had absolutely no technical expertise in the field which I took over in my postdoc at Harvard. So as I said, it was genomics, completely different. Okay, so and I wasn't new to this field, but I had an understanding of the literature of the genomics and the field, genome organization field. I was sort of inclined towards it in understanding how the problem which is in the field now. The DNA is very long in meters, but the cell is just few microns. Okay, it is 1 million times smaller than the length of, or 2 million times smaller than the length of the DNA. How you fit such a long thread into a very tiny cell? So it's a dimension problem, जिसको हम बोल सकते हैं कि dimension की problem है कि इतना बड़ा dimension है, इतनी छोटी सी cell में आप कैसे fit कर सकते हैं. So and इसकी physiological consequences then क्या है? So that is what I got intrigued into, and that was very, exciting field, and I switched to this to address, to learn. I cannot say address, so many people addressing the similar question or the same question. So to understand how this packaging, ये जो हमारा long DNA, how it gets packaged within this tiny cell. Okay, so that was the kind of the problem I started to look into, investigate when I joined at Harvard. And we made some advances. Small, I can say it's a small contribution. It's not we have solved something. No, that's not true. There are hundreds of labs across the globe who are working on the same problem. Okay, so and we showed that it is the proteins which help to some extent in packaging this long DNA into the tiny cell. Okay, so that was one of the major takeaways from my postdoc. We showed it by using a lot of technology, very high-end technologies which were available at Harvard. Okay, so that the proteins can help in this packaging of long DNA into the tiny cell. And what we did, we then changed this, we perturbed these proteins. We sort of perturbed their function. And then we saw that this packaging got perturbed. The packaging of this long DNA got perturbed. And other labs, as I said, there are other labs across the globe working. The other labs showed, in case of mouse, that the same perturbation can lead to a skeletal defect in the mouse. Okay, so although it has not been put in, you cannot put it in the humans, but you can show it in the mouse. And the lab, it was from Japan, when I was at Harvard. So they showed that same perturbation which we did in the cells, they did same perturbation in the mouse, and they showed that this can lead to the skeletal defects in the rib cage and one more vertebrae over here. It can lead to the extra sort of changes in the skeletal system of the mouse. So that was in nutshell sort of the research which I did in the postdoc, and obviously I gained a lot of technical expertise in the genomics field, and which I carried over here. And whatever I used to do there, except a couple of technologies, rest I think we can do now here in the University of Kashmir. All those experiments, okay, except a couple, but rest everything can be done here. So which I think is very satisfying to tell you that we can do something which is done in any very good lab across the globe.
So once you moved back home with a lot of expertise, a lot of knowledge upgrade and update as well. So what are you busy with in this lab?
So in this lab, I am continuing to address the same question. Okay, as I said, that we are trying to understand this long packaging into the long packaging of the long DNA in the tiny cell. So we are still addressing the same question. As I said, it's not something you start yesterday and you answer it tomorrow. Okay, so as I said in the beginning, when we started talking, what we know is the tip of the iceberg. There's still a long way to go, long way to go to understand this. So we started addressing this question again by looking at the other proteins. Okay, so how there are a lot of proteins, hundreds of proteins or thousands of proteins in the cell. We took another set of proteins and tried to see what is their role in packaging up this long DNA within the cell nucleus. Okay, so that is what we are sort of continuing, and we have some very good leads in showing that there are a few other proteins on which we are working. They play a major role in this packaging, and this packaging is something I can put it, understand it for a layman. Just like we have a thread, long thread, and now when we wrap it around in a spool, one goes. So it's long in meters, but we can then hold everything in the hand. So the spool, it's like a protein, and the thread is like the genome, is your DNA. And you solve the problem of dimensions, how you put a long thread into your hand of meters. So it is something similar, okay? I cannot say this is how it happens, but it is something similar to how we think the packaging happens, how proteins help in packaging this long DNA into the cell nucleus. And there are a lot of physiological consequences, as I was saying, of this packaging. So what if we change the packaging? As I said, we can develop the defects. So you might have seen people with fused digits. Someone has extra one. So what we learned few years back, not from my lab, but from the other labs, that it is because of a defect in the packaging of this DNA within the cell. So the packaging of that piece of DNA, which is responsible for developing our limbs, so at that place the packaging gets changed. With the result, we develop there's a developmental defect which results in the fusion of our digits, or sometimes an extra one is coming out. So, and in many cancers also, people have shown this packaging is changed. The packaging is changed, the function changes of the genome. Okay, it's a very sort of, I can say, very intricate, very complicated research question to be addressed. So in many cancers, in many developmental defects, we see this packaging is changed. If there's a change in the packaging, it might lead to a cancer in some cases, depending upon at which place in this genome the packaging has changed. Okay, if it changed at this place, it might result in a developmental defect in the limb. If it changes somewhere else, it might lead to another disease or a defect. So it can be inborn. Okay, we can inherit it, this defective packaging. So another aspect is that our genome is not isolated. It senses our environment. Okay, that is something, I mean, very interesting and very fascinating. It's not that I have my genome, I got it from my parents and that's it, it is their contribution, what I am. But no, it depends upon what I eat, where I live, what do I smell. Okay, so there's an interaction between our environment and our genome. For example, we can be in some environment which has toxic chemicals or something, which can change our genome, which can, what we call as mutations, which can lead to some mutations in our genome and in turn make us prone to some disease. Okay.
So can we simply say that the genome, the gene pool, is evolving with every generation?
So yes, definitely. I mean, I'm not an evolutionary biologist. Okay, let me put a disclaimer here. So I'm not an evolutionary biologist to answer exactly your question of what you are saying, whether our gene pool is evolving and it is getting better. Yes, obviously we know that in the nature there is natural selection. We have seen in case of the virus as it was evolving. This strain is now there, this is more virulent. This strain is there, it is more dangerous. What's that? It's adapting to the conditions, or we get antibiotic resistance against many bugs. Obviously it is changing, it is adapting. So but in our case, it is very, I don't know how to answer exactly in case of the humans. Okay, but yes, there might be some changes. How much they contribute, what are the time scales over which they can happen so that we can observe them. Okay, so it might take a very long time so that we observe. Okay, so it's completely an evolutionary sort of an topic to address upon. But yes, in other microorganisms which can replicate in very fast, we have definitely that.
When we say humans are adaptable, adjustable, flexible, I think we are somehow addressing the base of human life, that is genetics.
It depends upon, see, this changing our genetics is, as I said, it is the sequence of the 4 bases, which is very difficult to change. But what we can change is the epigenetics, okay, which is on top of the DNA, which is the proteins, okay, which help the DNA to perform some particular function, okay, and that is what is helping us to get adapted. Okay, for example, we eat regularly something, we need to produce more enzyme to digest it. that will be how we will get adjusted to something. Okay, and our body can sense that it can make the changes so that our cells make more and more of that enzyme. Okay, to adjust, to adapt to that change, that we are eating something more and we need more of that. But to change it at the level of the sequence of those 4 bases is not that easy. Okay, it's not that frequent, and even if it happens, it might not lead to the significant changes unless it happens at some very crucial places, which are very critical or very important for the normal physiological processes of a human being.
एजाज साहब, आप तो खुद साइंटिस्ट हैं, रिसर्च करते हैं, बट यू आल्सो मस्ट बी गाइडिंग अ ग्रुप ऑफ रिसर्चर्स, सो दे आर आल्सो वर्किंग ऑन द सेम सब्जेक्ट?
Yes, basically they are working on it. Okay, so I am just supervising right now, so I do a little bit of computational part myself, but they are actually my PhD students, and MSc students, so they work, actually they do experiments on the bench. Okay, so, and yes, obviously they work on the same question, and this is what I am talking is their experiments. Okay, and what we have learned from their experiments, and that's what sort of I am talking to you.
अभी तक सीरी से कितने PhDs निकले हैं?
So it is CIRI is new.
No, CIRI is new.
So it's just couple of years. It's still, I will say, in its embryonic stage. CIRI से अभी कोई नहीं PhD निकला है, but yes, from biotech, well, so वहाँ से बहुत सारे PhDs निकले हैं.
So how do you see CIRI 5 years from now?
So I would say, I mean, the CIRI will put this institution on the, I would not say only national, but God willing, on the international research map of the world.
डॉक्टर एजाज साहब, अपने बिजी शेड्यूल में इतना टाइम दिया, अपने एक्सपीरियंसेस शेयर हमारे साथ की, और एक उम्मीद जगा दी कि दुनिया, कायनात जितनी एक्सप्लोर होनी बाकी है, उसमें शायद हम भी कुछ न कुछ कर ही लेंगे. वी मे आल्सो कंट्रीब्यूट. Thank you, Dr. Sahab, for all these nice things and sharing this much of information.
Thank you.
Shukriya.
Thank you.
Assalamu alaikum.